Hi everyone, it's Dr. Hong again. Now today I am going to talk about acquired immune deficiency with a specific focus on HIV/AIDS. Now in this lecture, we will look at HIV/AIDS from an immunological disease perspective, and we will only briefly look at treatment for AIDS because I will cover that in future infectious disease pharmacology series. So let's get started. So here are a lot of the lecture objectives. Now basically we are going to look at how this HIV virus interact with the human immune system and how it affect the human immune system and how it make us sick. So that is the perspective of this whole lecture. Let's start with some statistics, which is the latest I can find in 2023. Now, the latest global HIV data was collected for 2021. 38.4 million people globally were living with HIV in 2021, and 1.5 million people became newly infected with HIV in 2021. Now, there are 650,000 people died from AIDS-related illness in that year. Globally, people who use injectable drugs remain at the top among all high-risk populations for contracting HIV. The infection pattern is different in the US. According to the US CDC data, in 2020, male-to-male sexual contact accounted for 68% of all new HIV diagnoses in the United States and dependent areas. Fortunately, the number of new global HIV cases each year peaked in 1996 and has been in decline since then. So, what is HIV/AIDS? The human immunodeficiency virus, HIV, is a viral pathogen that was first described in the early 1980s. AIDS is the clinical manifestation of infection by HIV, which causes a massive reduction in CD4 helper T cell. Now, CD4 cells are crucial for adaptive immunity, with cell-mediated activations of CD8 and macrophages and activations of B cells. The patients infected with HIV are unable to cope with innocuous flora, leading to opportunistic infections that can be recurrent or chronic. HIV-1 is the primary strain found in the developed countries and is believed to have transferred from a chimpanzee simian immunodeficiency virus. On the other hand, HIV-2 is endemic in West Africa and Asia and is less virulent, with slower progression. It is believed to have originated from a different type of monkey, the sooty mangabe monkeys. So, in case you're wondering, what are these monkeys looks like? So, on the left-hand side, we have chimpanzee, which is believed to have link to HIV-1. And the second picture on the right is the sooty mangabe monkey, which is believed to have relationship with HIV-2 strain. Now, under microscope, the left image is the HIV virion. The virion is an infectious vector that delivers viral genetic material to the host cells. HIV is an enveloped ssRNA retrovirus. The envelope is composed of lipid derived from the host cell's membrane and glycoproteins coded for by the viral genome. One such glycoprotein is GP120 that can bind tightly to the human CD4 receptor. Another glycoprotein is GP41, which mediates the fusion of the viral envelope with the plasma membrane. Together, these two envelope glycoproteins play a crucial role in the entry and infection of the host cells by HIV. The HIV virus has a nucleoprotein core or nucleocapsid, which houses its genetic material. The genome of HIV consists of two single-stranded RNA molecules, which are essential for the virus's replication and transmission. Within the nucleocapsid, HIV also contains several enzymes that are crucial for its viral cycle. One such enzyme is protease, which helps the virus to break down and assemble its structural proteins. Another enzyme is reverse transcriptase, which converts the viral RNA genome into DNA during the early stages of infection. And finally, HIV contains an enzyme called integrase, which is responsible for inserting the viral DNA into whole cells' genome. HIV infects dendritic cells, macrophages, and helper T cell, which are vulnerable to infection because they express the CD4 receptor that the virus utilizes for entry. The GP120 glycoprotein of the virus binds to the CD4 receptor, but it also requires the binding of a co-receptor to facilitate viral entry into the host cell. Two co-receptors have been identified: CCR5, which is expressed on dendritic cells, macrophages, and T cells, and CXCR4, which is expressed on activated T cells only. Now, after binding of the co-receptor, the GP41 envelope glycoprotein mediates the fusion of the viral envelope with the plasma membrane of the host cell, allowing the virus to enter and infect the cell. Again, HIV is a virus that infects CD4 T cells, macrophages, and dendritic cells. And there are two main variants of HIV: macrophage tropic and lymphocyte tropic. The macrophage tropic variant of HIV infects macrophages, dendritic cells, and binds to the CCR5 receptor. Only modest level of CD4 binding are required for this variant, which is commonly associated with the primary stage of disease. In other words, the first cells to be infected are actually macrophages and dendritic cells. On the other hand, the lymphocyte tropic variant of HIV infects CD4 T helper cells and binds to the CXCR4 receptor. High level of CD4 expression, as found on activated T cells, are required for this variant. The viral phenotype switches to lymphocyte tropic late in infection, leading to a rapid decline in CD4 T cell counts and progression to AIDS. Now let's look at the HIV viral cycle in great detail. The HIV infection begins with the binding of the virion to the CD4 receptor on target cells. The GP120 glycoprotein on virion binds to the CD4 receptor as well as to the co-receptor CCR5 or CXCR4. The GP41 glycoprotein then mediates fusion of the viral envelope with the plasma membrane of the host cell, releasing the viral core into the cytoplasm and releasing the RNA genome. The virus uses the enzyme reverse transcriptase to make a complementary DNA or cDNA copy from the RNA genome. The cDNA migrates to the nucleus and is inserted into the whole cell genome by viral integrase. The integration creates a provirus that is found in infected CD4 T cells, dendritic cells, and macrophages. Meanwhile, the virion can be found in bodily fluids such as blood, semen, vaginal fluid, and breast milk. When a CD4 T cell is activated by the presence of a pathogen, it produces the transcription factor NF-kappa B, which initiates the transcription of the viral DNA of HIV that has been integrated into the host cell genome during the provirus stage. The early proteins TAT and RAF are then produced. TAT amplifies transcription of the viral cDNA, and RAF, on the other hand, controls the supply of viral RNA to the cytoplasm and the extent to which that RNA is spliced. The late proteins GAG, POL, and ENV are assembled into an infectious virion, which butts into the plasma membrane of the host cells and allows the infectious virion to leave the host cells and infect new cells. HIV infection occurs primarily through the transfer of bodily fluids, such as blood, semen, vaginal fluid, or milk. The virus can be transmitted via virions in these fluids or through the provirus carried in infected CD4 T cells, dendritic cells, and macrophages. Immediately after infection, the patient may be asymptomatic or experience a transient flu-like illness. As the virus becomes abundant in the peripheral blood, the circulating numbers of CD4 T cells drop, resulting in acute viremia and the development of an HIV-specific immune response. This response leads to the formation of anti-HIV antibodies and activations of cytotoxic CD8 T cells, which reduce viral load and increase the CD4 T cell count. The presence of detectable levels of anti-HIV antibodies in serum is referred to as seroconversion. HIV infection can cause symptoms in around 8 to 10 years if left untreated. The virus goes through an initial phase of infection followed by an asymptomatic period known as clinical latency. During this time, there is a gradual decrease in T cell numbers, particularly CD4 T cells. As the CD4 T cell count drops, the immune system becomes increasingly weakened, and the individual becomes more susceptible to opportunistic infections and cancers. Eventually, the CD4 T cells drop into a concentration below the level necessary to mount an immune response, marking an onset of AIDS. In HIV infection, lymphoid tissue serves as a major reservoir for the virus. The key lymphoid tissue involved are lymph nodes, germinal centers, follicle dendritic cells, subcapular macrophages, and the T cell area. Now, these tissues contain infected monocytes, macrophages, dendritic cells, and CD4 T cells that produce and store infectious virions. The majority of the virus production occurs in the T cell area, with 95% of the virus produced by CD4 T cell. Now, the germinal centers serve as a storage site for infectious virion, and FDC can harbor virus for extended period of time. Subcapsular macrophages also serve as a site of viral replication. In addition to lymphoid tissue, wandering macrophages can infect other parts of the body, including the brain microglial cells, leading to neurological complications. Thus, targeting viral replication in lymphoid tissue is critical for the effective management of HIV infection. This is a classical illustration to illustrate the adaptive immune response to HIV limits the effect of infection. Now, during the course of infection, the levels of HIV virions and other components of the adaptive immune system in the blood plasma changes. Now, early in infection, while the adaptive immune system response is being activated, the virus reaches high level indicated in the red line, with the production of HIV-specific antibodies indicated in the blue lines and HIV-specific cytotoxic T cells with the yellow line. The virus is kept at a low level but is not eliminated. When the destruction of CD4 T cells outstrips their rate of renewal, adaptive immunity gradually collapses and virus levels increase again. The HIV virus is a challenging pathogen to eliminate due to its unique characteristics. Unlike other pathogens, HIV is a retrovirus, which makes it even more error-prone during replication and transcription. Mutations occur rapidly, resulting in different strains isolated from each individual patient. This high variability foils the immune response as the immune system fails to recognize the virus. Additionally, HIV is prone to errors in replication, making it difficult to target and develop effective treatment. These characteristics make vaccine development almost impossible, and the treatment is often prolonged and challenging. Now let's look at a special HIV patient case which was published in the journal Nature in 2019. In this report, a patient with HIV has experienced sustained remission of the HIV-1 virus following a bone marrow transplant. Now, in a case study involving a bone marrow transplant, a patient who was positive for HIV-1 and Hodgkin's lymphoma received hematopoietic stem cells from a donor who expressed a homozygous recessive mutation of CCR5. Now, CCR5, again, is a co-receptor that HIV-1 uses to enter CD4 T cells. And by having this mutation, the donor's cells were resistant to HIV-1 infection. After the transplant, the patient experienced a remission for 18 months and showed a decrease in viral load and antibodies for HIV in their blood. Now, this was likely due to the replacement of the patient's immune system with that of the donor, which lacked HIV-1 producing CD4 T cells. Now, while this case study showed a promise for a potential cure for HIV-1, the risks associated with bone marrow transplant and the rarity of finding a suitable donor with this type of mutation make this option unfeasible for the general population. One of the biggest problems or challenges with HIV infections is it can lead to immunodeficiency and deaths from opportunistic infections. Now, CD4 T cell death is a major factor in the progression of HIV to AIDS, and there are several mechanisms by which CD4 T cells can die, including lysis and apoptosis of infected cells, as well as CD8 T cell promoted death. As the number of CD4 T cells declines, the immune system becomes less effective and the body becomes more susceptible to infections by commensal organisms and opportunistic pathogens. This increased susceptibility to infections, and which is a hallmark of the progression to AIDS. Opportunistic pathogens can increase the risk of deaths of individuals with AIDS. Fungi, such as Candida species, can cause oral thrush, a common opportunistic infection in people with AIDS. Bacteria, like Mycobacterium tuberculosis, can cause severe lung infection in AIDS patients. Reactivations of viruses like herpes simplex can also occur in individuals with AIDS, leading to painful lesions on the skin and mucosal membranes. In addition to these infections, people with AIDS are at increased risk of developing blood cancers such as B-cell lymphoma. Now, these opportunistic pathogens can cause severe illness and even death in individuals with AIDS, highlighting the importance of proper management and treatment of the disease. And antiviral therapy, or ART, has been a game changer in the treatment of HIV/AIDS. The highly active antiretroviral therapy, or HAART, introduced in 1996, is a combination therapy that involves administering 3 to 4 antiretroviral agents in combination cocktails. Now, these cocktails typically include 2 nucleoside or nucleotide reverse transcriptase inhibitors, NRTIs, and one other agent, which may be a non-nucleoside reverse transcriptase inhibitor, an NRTI, a protease inhibitor, a PI, fusion inhibitor, integrase inhibitor, or CCL5 antagonist. Now, each of these agents works in a different way to reduce the viral load by inhibiting viral replication or entry into host cells. And by combining multiple agents with different mechanisms of action, HAART can prevent the virus from replicating and decreasing the likelihood of developing drug resistance. This approach has significantly improved the prognosis for individuals living with HIV/AIDS and has enabled many to live long and healthy lives. Now, I will discuss more antiviral therapy in my future pharmacology series. So that is everything for this lecture. So stay tuned and I will talk to you next time. Take care. Bye.